Metabolism of lipids consists of biosynthesis and oxidation of lipids, or anabolism and catabolism of lipids. In this lecture, we will examine biosynthesis of lipids. According to classification based on biosynthetic pathway, fatty acids are polyketides. Polyketides are formed by coupling of fatty acids in condensation reaction. 2-carbon unit, that would be acetic acid, so condensation of acetic acid to give a polyketide. Polyketides are fatty acids, prostaglandins, and some aromatic compounds. But actually polyketides are not formed from acetic acid, but from its derivative, acetyl coenzyme A. Acetyl coenzyme A is product of decarboxylation of pyruvate, and pyruvate is the end product of glycolysis. In repeating Claisen condensation reactions, acetyl coenzyme A molecules form a polyketide chain. In living organisms, the reaction to synthesize acetoacetyl coenzyme A and ultimately fatty acid proceeds in more complex and more efficient pathway. So it does not proceed by Claisen condensation. Instead, acetyl coenzyme A is first converted to malonyl coenzyme A. Now, malonyl coenzyme A is synthesized in carboxylation reaction of acetyl coenzyme A. The reaction is enzyme catalyzed with biotin, sometimes called a vitamin B7 as coenzyme. Reagent is carbon dioxide in form of its bicarbonate anion, and one equivalent of ATP is used up in the reaction. Initially, ATP reacts with bicarbonate to give a mixed anhydride of phosphoric and carbonic acids. The mixed anhydride reacts with biotin enzyme complex to give carboxylated biotin, and then finally carboxylated biotin complex transfers the carboxyl group to acetyl coenzyme A enolate to give malonyl coenzyme A. In animals, biosynthesis of fatty acids is catalyzed by a single multifunctional enzyme, and that's the fatty acid synthase. In bacteria and plants, a series of different enzymes is involved. Each malonyl coenzyme A and acetyl coenzyme A are converted into corresponding thioesters. So it's transesterification reaction. And they are involved in biosynthesis of fatty acids as thioesters attached to acyl carrier proteins, or ACP. Now a reaction is Claisen condensation, and that Claisen condensation gives the acetoacetyl acyl carrier protein as the product. Claisen condensation now proceeds with simultaneous decarboxylation. Decarboxylation is loss of carbon dioxide, and so it's a highly favorable process. So malonate that was formed in the previous step by a carboxylation reaction has additional carbon, and now loss of that carbon makes the process highly favorable. So it is still only a 2-carbon unit in the form of acetyl coenzyme A that is incorporated at the end of the fat into the fatty acid. So 1 carbon was added in carboxylation reaction, and then it was lost in decarboxylation reaction. And so the overall ATP molecule was used up to introduce that carbon in carboxylation reaction. And so actually the whole process means that at the expense of 1 molecule of ATP, the entire process has been made favorable. This is shown here. Malonyl coenzyme A and acetyl coenzyme A are transferred to acyl carrier proteins, and in Claisen condensation they give acetoacetyl acyl carrier protein. In the next step, NADPH reduction of acetoacetyl acyl carrier protein gives the corresponding beta hydroxy ester. Elimination of water from it gives the corresponding trans alpha beta unsaturated ACP ester. So a trans compound is only a short-lived intermediate. and it never leaves enzyme complex. It is immediately converted to a saturated ester in the next step by NADPH reduction. Resulting fatty acid ACP thioester can repeat cycle with another molecule of malonyl ACP to give fatty acid with 2 additional carbon atoms. And cycle keeps on continuing. After each cycle, fatty acid has 2 more carbon atoms. At the end of biosynthesis, fatty acid is released either as fatty acid coenzyme A or as a free fatty acid. The chain length of a fatty acid is controlled by the enzymes that catalyze release of fatty acid from the fatty acid ACP complex, and those enzymes recognize the length. So once chain length of fatty acid reaches certain length, then enzyme recognizes its correct length and releases fatty acid from ACP complex and ends the biosynthesis. Unsaturated fatty acids are synthesized from saturated fatty acids. In animals, fatty acids that are present in fats are mainly saturated. Fats from fish and plants are mainly unsaturated. There are several biochemical pathways for synthesis of unsaturated fatty acids, and all of them involve dehydrogenation of the corresponding saturated fatty acids. First enzyme, the most important enzyme that is involved in this process is delta-9 desaturase, and NADP and oxygen are the stoichiometric reagents. Substrates for dehydrogenation are coenzyme A esters in animal and fungi and ACP esters of fatty acids in plants. As delta-9 saturase is involved, initially produced monounsaturated carboxylic acid is delta-9 unsaturated fatty acid. The enzyme is named delta-9 desaturase because it introduces double bond between carbons 9 and 10. So it introduces double bond 9. So an example given here, shown here, is that dehydrogenation of stearic acid gives oleic acid. And then additional dehydrogenation can introduce new double bonds to produce polyunsaturated fatty acids. All of the double bonds in naturally occurring fatty acids are cis or Z, and if fatty acids are polyunsaturated, double bonds are separated by methylene group, CH2 group. After delta-9 double bond is introduced, then next double bond can be introduced between delta-9 double bond and the methyl terminus, or delta-9 double bond and carboxyl terminus. In mammals, additional double bond is introduced towards the methyl terminus. In mammals, actually additional double bond is introduced towards the carboxyl terminus, while in plants towards methyl terminus. And so linoleic acid, shown here, is precursor of eicosanoids. It has double bond towards the methyl terminus. It has to be obtained from the diet. Actually, linoleic acid is precursor of arachidonic acid, which in turn is precursor of eicosanoids. So linoleic acid is an example of an essential fatty acid. Arachidonic acid, which we have already encountered several times in this course, is precursor for biosynthesis of eicosanoids. It can be obtained from the diet, such as pork meat, fat, eggs, fish, or it can be synthesized from polyunsaturated fatty acids. In the Western diet, the most common biosynthetic precursor of arachidonic acid is linoleic acid, which is present in plant oils. And usually our diet, Western diet, contains sufficient amount of plant oils that biosynthesis of arachidonic acid is not a problem. Now, arachidonic acid is conditionally essential fatty acid, which means that if the diet does not contain sufficient linoleic acid, for example, if person consumes a diet that does not contain vegetables, fruits, or nuts and only contains, let's say, poultry meat, then arachidonic acid becomes essential fatty acid and must be obtained from the diet. In humans and most mammals, biosynthesis of arachidonic acid occurs in the endoplasmatic reticulum, and biosynthesis involves two desaturation steps and an elongation step. So they are shown on the next slide. Starting with linoleic acid enzyme, D6 desaturase gives gamma linolenic acid, GLA. So delta 6 desaturase means that it introduces double bond between carbons 6 and 7, double bond 6. This step is controlled so that amount of arachidonic acid does not depend on the amount of available linoleic acid or suitable precursor. Only the amount of arachidonic acid that is needed is produced. So consumption of large amount of linoleic acid will not result in biosynthesis of large amount of arachidonic acid. Gamma linolenic acid undergoes chain elongation catalyzed by elongase 5 enzyme to give dihomogamma linolenic acid, DGLA. And so, as a result of elongation, C18 carboxylic acid is converted into C20 carboxylic acid. And so note now how double bonds have shifted, because two carbon atoms that have been added are carbon atoms 1 and 2. And so original double bonds have shifted. So what used to be 6-7 double bond or double bond 6 is now double bond 8. What used to be double bond 9 is now double bond 11. And what used to be double bond 12 is double bond 14. Which means that double bond 11, even though it's double bond 11, its introduction has been catalyzed originally by delta 9 desaturase enzyme. Double bond 8, its introduction was catalyzed by delta 6 desaturate enzyme. That's very important. And finally, enzyme delta 5 desaturase converts dihomogamma linolenic acid into arachidonic acid by introducing remaining fourth double bond in position 5. And so the whole biosynthesis proceeds from linoleic acids to gamma linolenic acids, dihomogamma linolenic acid, and finally arachidonic acid. The actual biosynthesis is shown here and involves acetyl coenzyme A derivatives of the acids, and also process is a little bit more complex. If you wish, you can pause the video here and examine process in more detail. Cats are not able to convert linolenic acid to arachidonic acid. So for them, arachidonic acid is an essential fatty acid and they must obtain it from the diet. So for that reason, a vegetarian diet is not suitable for cats. While that may sound to be disadvantageous, it's actually an evolutionary advantage, because simply by eating arachidonic acid, cats and similar animals for whom that's essential fatty acid actually have evolutionary advantage, because they don't have to expend energy on biosynthesizing arachidonic acid. They simply obtain from food, they eat something that somebody else, that another organism expended energy on consuming it. That's evolutionary advantage as long as there is readily available food source of it, in this case food source of arachidonic acid. Otherwise, if not, then of course those who can biosynthesize arachidonic acid have advantage. Biosynthesis of phosphatidates is the first stage in synthesis of complex lipids. Suitable precursors are glycerol or dihydroxyacetone. Dihydroxyacetone is product of glycolysis, and as product of glycolysis, it leaves glycolysis as dihydroxyacetone phosphate. So it has to be reduced by NADH to glycerol 3-phosphate, and then it can enter synthesis of phosphatidates. Otherwise, if glycerol is precursor, then it is phosphorylated into glycerol 3-phosphate. So either way, glycerol 3-phosphate then is precursor for further synthesis of phosphatidates. So fatty acids are first activated by forming either coenzyme A or acyl carrier protein thioesters in a reaction that consumes ATP. Next, in reaction catalyzed by an acyl transferase, ester group on carbon 1 of glycerol 3-phosphate is introduced. So that's the first group that is introduced, and then and that is usually saturated fatty acid such as stearic acid. In humans, it's actually usually palmitic acid. In the next step, which is catalyzed by different acyl transferase, an ester group on carbon 2 is introduced, and that's usually an unsaturated fatty acid such as oleic acid. It could be also linoleic or arachidonic acid. Acyl transferases are not specific for particular chain length, but are specific for carbon atom carbon 1 or carbon 2 of glycerol 3-phosphate. And so this yields molecule of phosphatidate. It is precursor for biosynthesis of membrane lipids such as phospholipids and triacylglycerols. Triacylglycerols are long-term energy storage. So this completes our examination of biosynthesis of lipids.